And at the same time, for this tracer, of course, it's this demonstrated clinical use and the cell kind of the changing. However, and this tracer have a kind of irreversible collapse, means and the compound binding and the kind of you cannot the kind of the displacement. And later, a lot of many group also develop different of kind of tracer and they try to image in the Sigma-1. This is a several tracer already used in clinical, but particularly F-18 developed by Stanford. And all of these tracers have a similar kind of a program and the easy kind of irreversible binding or kind of metabolized concept. And we know developer PETA tracer to send why you have to use which isotope. Basically, we talk about they already have a carbon in there, have an F-18 tracer. Actually, clinical use beta isotope, also potential O15 and also nitrogen 60. And here is a half-life. Actually, the half-life makes it significant. You think about it, if you want to get a tracer half-life is 2 minutes, you need a first make it a quality control center for human study. That will be very challenging. Even carbon 11 tracer, you can only make on the cyclotron on the clinical side. You cannot say I make the tracer, then I send it to different clinical center, then do the study. And if you compare F-18 get 110 minutes, you make the dose, and you can kind of sitting around like 3 to 4 hours and supply different clinical center and for beta study. Other isotopes, because they report also potential could be a beta tracer, but you can see actually their half-life got it. And therefore develop the Sigma tracer. At the beginning, actually my research focused on developing the vesicle acetylcholine transporter, and this is also a hard topic. And at the beginning, the worker started from 1980, never get a tracer. So you can see this is a VSHP kind of pharmacologic. This is a Sigma pharmacologic. And we optimize this kind of structure. Eventually, and each group get this one, move to clinical. I think in 2014, we kind of also work on this kind of modification. Eventually, we just introduce a carbonyl group, and here we get the compound and the back and move to clinical. Now this tracer already kind of finished more than 300 patients study, and the clinical physician and they saw this tracer kind of have a clinical favorable pharmacologic. And at the same time, we found that and the VSHP compound, and they have a program. They actually some of the compound have a sigma binding potency. Okay, and so we kind of summarize all of this compound, and we reported that. And in 2011, we can see some of the VSHP compound, of course, it's potent, but some of this kind of skeleton you really get a high sigma affinity. For example, right here, okay. And this is the leading compound is at the time of the carbon 11 SF458. Then it's trigger us, okay, we can kind of design and modify the structure, try to get some kind of Sigma-1 specific. Okay, we reported that here 23 ligand, and actually 4 of them have a very high potency for Sigma-1 with Sigma-2 and also VSHP. And we know the Sigma binding have a stereo selectivity, so we do the structure, the CRISPR structure, we identify and which isomer and the binding more potent, and eventually we identify these two compounds and they have a very high binding potency for Sigma-1, also high selectivity for Sigma-1 with Sigma-2, and also other dopamine receptor and also other neural receptors. Okay. And they come out actually this is 2 years ago because the Alzheimer diseases they get more and more attention. And the Sigma-1 inhibitor treated Alzheimer diseases they get a clinical trial and they saw it promise. Then we revised our compound. First, we kind of try to choose this 6 compound to the carbon 11 labeling for proof of conception. And we do the modeling dark study. We found that they actually binding with the glycol, the Ru-172, and also for the other three isomer, they involve different hydrogen bond. Okay. And then we kind of do the kind of study in the rodent and also the non-human primate. We can see and the compound 7 and also 11. We do the broken study use haloperidol. We can see and also use the other one, TS318. Both can block the uptake. It can block means it's can compete with the radiotracer, which demonstrated the compound specific binding with Sigma-1. Okay. At the same time, we do the non-human primate. We can see and the tracer can go to the non-human primate brain very well. Okay. And the data and they come out the last 2 or 3 years. Of course, our goal is get an F-18 tracer. Then we identify this kind of racemic mixture after color resolution, get a minus plus, and all of this kind of easy and isomer or kind of racemic mixture potent. Then we labeling all of this compound and do the in vivo validation in animal model, in rodent, and also in non-human primate. And then here is the first step. We can see this compound in the biodistribution, which means you inject the tracer to the animal, then you can kind of utilize it in different time. Then you get all this kind of tissue distribution. See if first if the tracer actually get it into the brain, and the second if you really get the bone increase, because if you get a bone increase, that means the compound get into the vivo get a defloration. Okay. For this compound, at least we didn't say that means the compound don't have a defloration. At the same time, the brain uptake also show very good. And we also do the autoradiographic study. Autoradiographic study basically is use the radiotracer incubated with the brain tissue, the same tissue, also adjust the tissue. And you do the kind of immunostaining, demonstrate that the compound actually binding with the Sigma receptor. This is immunostaining use it for the Sigma-1, okay? And this is the brain adolescent. At the same time, in vitro, we can do the broken study incubated with a coded compound and binding with Sigma-1, see if the potent, if the activity can get it kind of the block, okay? And the cell is broken well, and also the distribution of the tracer with the Sigma-1 receptor match very well. And then we can see in the brain, actually the potassium and also product the high and the similar copper, the copper colossum have a kind of relatively low the compound specifically binding with Sigma-1. Okay. And then we did kind of a broken study. Basically, you inject the tracer, also inject the coded compound in animal model. First, we can see, okay, this is the baseline. This is a use a broken agent, the haloperidol, and this is use kind of the TS318 and the Sigma kind of analog. All of this kind of show this several compound we can use a haloperidol and the significant block in the rodent brain, which means all of this several tracer actually binding with Sigma-1. Here is the image. You can see that the top one, the top one is kind of like then the block study. Okay, use different compound for block. Okay. And then we actually move forward. We say we want to image the Alzheimer diseases. Then we use the three multiple and the transgenic AD mouse and do the study. And we can see the AD mouse and compare with the control. Actually, we can see and the AD mouse get the Sigma-1 get decreased. Okay, and compare with the control, which. Yeah, which means the tracer can actually measure the Sigma-1 kind of expression, okay, in the brain. And then after that, we did a kind of conform the Sigma-1 expression in the baseline and also in the AD brain. We can see in the AD brain compared with the baseline, and the Sigma-1 get a kind of decrease. Okay, and this is the quantification show how much decrease. Okay, and then we didn't come back and do the kind of and the monkey study demonstrate that. Of course, we kind of have 6 compounds. We want to see, okay, which of them and they go to the brain have a good kinetics because we criticized previous tracer that kind of is a risk for biting, which means the biting like this or continue increase. In that case, you really have to reach the equilibrium. You cannot use for target engagement. We really want the cells to tracer get into the brain and then can wash out like this one. And why you need a non-human primate? Because primate brain is a physical and the physiology close the human brain. If the tracer can show in the non-human primate kind of and a good property, which means it maybe can transfer to human. Okay, and then we can see the tracer go to the brain well and also wash out very well, particularly the compound F-18 CT minus, okay? And then come out with the metabolites. And then how you do the metabolites, we cannot kind of do the monkey brain tissue or human brain tissue. What we do, we can kind of do the non-human primate plasma. Basically, after you inject the tracer, in different time, you take the plasma out from the animal, the monkey, during the beta scan. Then you kind of do the process injected to HPLC, and basically you can see in different time point how many the parents compounds still retaining and what the metabolizer and the concentration. And of course, and here you also want the HPLC because the compound if it's more lipophilic than the parent's compound, which means it's a potential close to the blood-brain barrier. And what you really want to demonstrate that if I metabolize, because every compound goes to the vivo where metabolized, you want to demonstrate that no lipophilic metabolizer can cross the blood-brain barrier and have some hydrophilic metabolite. We don't think it's worth go to the blood-brain barrier. Because for non-human primate, you cannot take the non-human primate for brain tissue to the metabolize analysis because it's also very expensive. And each non-human primate costs like 10,000 US dollars. And then you can do it in the rats. In the rats, you can inject the tracer, go to the rats. Then you utilize the rats in different times. We take the brain tissue and then do the metabolize analysis for the brain sample. And then you can detect that if you get the metabolize get it into the brain or not. We demonstrated the compensating have a good metabolite stability and also no metabolite can cross the blood-brain barrier get into the brain. And then we come back, okay, and literature reported a lot of controversy. One receptor, they say maybe the age dependent, and it means younger one, older one, or middle age, and maybe have a different expression. And here we just choose the kind of a different age of the animal. You can see here is the age. And however, we also see in a younger animal, seem have a high uptake compared with and the relatively older animal. Because this is the easy group only get 1 or 2 kind of animal. And we still want to do more animal demonstrate if really is dependent. Okay. And then is my summary because we develop a class of compound. We evaluate them. We think that this is the best compound. And the cell promise our next step is to do a toxicity study, then get an FDA approval for human use, and then perform a clinical trial in Alzheimer disease. And I'd like to thank you all of this individual from our facility and also all of these people and help my career and also my group and also our cytotron facility around the administration.